It is my distinct pleasure to introduce and welcome Professor Anna Bento from Indiana University, Bloomington. Professor Bento is a disease ecologist with a focus on ecology and evolution at the intersection with eco-epidemiology, and is focused on infectious diseases. She wears many hats and has appointments in epidemiology and biostatistics in the School of Public Health and in the Evolution and Ecology Department in Bloomington. After earning her undergraduate and PhD degrees in evolution and ecology at the Imperial College London, she conducted postdoctoral research at the University of Georgia, collaborating with the CDC, studying the ecology and evolution of vaccine-preventable childhood diseases. She has also further worked on infectious disease dynamics within livestock herds, essential to the human food and nutrition supply chain. So we're so excited to host this talk because Professor Bento is addressing human health both directly in understanding childhood vaccine preventable diseases, but also through indirectly our food supply chain, which is quite important depending on where you live in the world in very, very, very big ways. And so she just is a fantastic scholar who's bringing together foundational evolutionary principles with their practical applications to human health, and trains her mentees through this lens and really situates her work at how do we leverage and harness evolutionary ecological principles in service to understanding things that are going to be directly translatable to improving human health. So just right in line with our center's interests. So please join me in welcoming Professor Bento and her talk today on integrative Bayesian approaches to assess epidemiological and evolutionary impacts on disease dynamics. Welcome.
Hi, all. Thank you so much for having me. And thank you, Katie, for such a kind introduction. So as Katie said, I particularly focus on eco-evolutionary impacts of anthropogenic drivers, for which I tend to think of vaccination as being one of them. And I look at those anthropogenic drivers and how they affect disease transmission, both in humans and other animals. And what I tend to focus a lot is on demographic heterogeneity and structure in populations. So when you think of infectious diseases, of course, now all the rage is COVID-19, but I thought I'd give you a break today. And so I'm going to be discussing a study that has been ongoing for a while on one of my favorite study systems in humans, whooping cough, also called pertussis, that uses parallel data streams to address both ecological and evolutionary patterns of re-emerging pathogens. All right, sorry, I was having trouble passing through the slides. So in the age of Anthropocene, one of our biggest challenges is the continuing global impact of infectious diseases. And this is no longer a statement that anyone actually disagrees. But it's also clear from COVID-19 at this point in time that control strategies such as vaccine programs, such as a bad use of antimicrobial interventions and so on, while they continue to be completely successful in preventing and saving lives, if they are misused, they actually have the ability to suppress pathogen fitness so that they can actually impose evolutionary pressures on pathogen populations that could cause selection for antigenic replacement, resistance, and increased virulence. So in this plot here, which is data from the CDC for the US, you actually see in orange when the introduction of the vaccine was. And for the most part, you actually see an immediate reduction in prevalence as we go down in time of these diseases. However, what we see, for instance, with pertussis, with measles, with mumps, and so on, we actually see that lately we're starting to see a bounce back, a reemergence of these diseases that should have been already controlled. And this is the kind of thing that I'm very interested in, this idea of why are some diseases re-emerging in a few populations, but not overall. So there are several examples of those selective pressures that I mentioned. So increased virulence was one of the things that I mentioned earlier. And a very good example of that is myxomavirus in rabbits in Australia, which is a very interesting case study, but also Merrick's disease, which is actually one of my favorite diseases, even though I don't study it. So the idea here is that immunization can actually make once rare or non-existing genetic variants of these pathogens that are circulating actually become more prevalent. So in the case of Merrick's disease, we had a vaccine that was introduced in 1970s, and when the disease was killing basically the entire flock of poultry. But what happened was that that vaccine was actually not perfect. It was a leaky vaccine. And so what we started to see was the actually propping up of some variants that were escaping the vaccine and were actually causing mortality in the non-vaccinated populations. So this is the biggest problem with the leaky vaccines, is that they actually enable pathogens to replicate unchecked. And so while they're protecting the hosts from illness and death, we are now removing the costs associated with increased virulence over time. And with these kinds of leaky vaccines, the pathogen actually evolves to become deadlier to unvaccinated hosts because it can actually reap the benefits of virulence without the costs. So another good example of how control strategies sometimes have unintended consequences is antimicrobial resistance. So here we see that in the darker lines, we have when resistance starts to evolve, and then it takes a while for some of these to completely be resistant to the drugs. So this is where we see the lighter color, either of the yellow or the blue. So the idea is that pathogens can rapidly or not so rapidly evolve and adapt in response to changes in their environment. In this case is the introduction of an antimicrobial drug for control. And these external influences happen when they are improperly used or used for too long. So this is another example. So the other one that is incredibly well studied and well known is antigenic drift in influenza. So here is a slightly different story in it, that a new flu vaccine needs to be developed every year because influenza viruses naturally mutate very quickly. So antigenic changes accumulate. The antibodies created against the older viruses no longer recognize the newer viruses, and thus we have to get vaccinated every year because of these genetic changes can result in a virus that is differently antigenically in terms of properties, therefore the need for revaccination. So this is like my PSA, get vaccinated every year. But also there is some starting to create some sort of like parallel concepts as we start developing vaccines for COVID-19. We might find ourselves in a situation that could potentially be similar in the years to come, but of course it's still too soon to truly understand that. So I mentioned that today we're going to talk about the reemergence of pathogens where there is incomplete vaccination and why that does not paint a picture. And I just wanted to tell you why this is still important, even though we are in a pandemic. So there was a study that came out from the CDC a few months ago that actually showed that during the lockdown period, there was a massive decrease in MMR coverage in the US. So we actually see a massive drop in vaccines. And what we're seeing at the moment, which is not shown here, but I acquired those data, is that there hasn't been a bounce back in terms of coverage. So likely we will have cohorts of individuals in the years to come that actually did not get vaccinated or will have to take the vaccine much later, which will result in a very different immune profile in our population for the years to come. So understanding this mosaic of populations will be very important, not just in terms of disease dynamics, but also evolutionary pressures, as we'll have different populations with different profiles. So today's talk is, as I mentioned before, is going to be focusing on pertussis and trying to interrogate whether we can ascertain if the reemergence of pertussis might be actually vaccine-driven evolution in action. And so this talk is more on the pathogens point of view rather than a straight up population dynamics kind of approach. So a little bit of background on pertussis, because I'm assuming not all of you will know what pertussis is. So pertussis is primarily caused by the bacterium Bordetella pertussis, and it's highly contagious, which means that in the pre-vaccine era, the mean age of infection was between 4 and 6 years of age. So people were actually getting infected very early on. And because of its very simple natural history, life history, it was actually once a candidate for eradication after the vaccine was introduced. However, if the story was that simple, I wouldn't be telling you this story today. And there was indeed a plot twist. So in some countries where vaccine coverage is very high, so the ones that I'm highlighting here in red, we're actually seeing now an increase in prevalence in these countries. However, the causes for this remain highly contentious. Pathogen adaptation has now recently been suggested, trying to understand if it works through different types of mechanisms. So it could be immune evasion, where there is antigenic divergence between the vaccine strains and the circulating strains, so a similar picture to the influenza story, or if it's emergence of strains that are more virulent, similar to the Marek story. So I wanted to give you a little bit of a piqued interest to let you know that, of course, this is happening all throughout the US, but it's also a problem in Arizona. So in blue, we see the time series of pertussis in Arizona from the pre-vaccine era to very recently. And in red, we actually have I have the Tempe data. So these were data that were sent to me by the Project TICO and also the CDC. And what you see is that even though, of course, after the introduction of vaccine in the late 1940s, where we see this decrease in incidence, but also this increase in periodicity between outbreaks, in the late 90s, we're now starting to see an emergence of pertussis in these populations. And some of these outbreaks are of the same magnitude of the pre-vaccine era. So what we can't really understand just yet is whether incomplete vaccination tells us the full story or not, because particularly in Arizona, vaccine uptake is incredibly high. So my story today is going to have two focuses, but the questions are the following. So I'm interested in understanding whether for the bacterium population, whether there are traits that are changing, if vaccine changes are driving bacterial selection, if I can find that signal, and how does genetic diversity particularly actually translates into changes in transmission. So with that, I'm going to take the talk into two parts. The first one is a data-driven Bayesian coalescent type model, so a more evolutionary approach, where I'm looking to understand if I can identify covariates of selection. And the second part, we're going to switch gears a little bit and try to quantify contributions of different immune profiles of the population to the transmission dynamics. So this is going to be a mathematical simulation type model. So I mentioned that we were going to focus on pertussis, but today's study case is on pertussis in the Netherlands. So why Netherlands? I'm giving a talk in the US, I'm from Portugal, so why did I choose the Netherlands? So the Netherlands is one of the countries where pertussis is re-emerging. In addition to that, they actually have incredibly detailed parallel records, both on epidemiological data, but also on isolate sequence data. And it's so small that every type of policy that is introduced, vaccine changes and so on, is done incredibly homogeneously, or as homogeneous as we can do it, such that we can truly evaluate changes at the national level. So here we have pertussis in the Netherlands. So on the left, we have annual cases, and on the right, we have vaccine coverage. We know that notifications, so compulsory notifications of pertussis started in the mid-70s. So of course, there were cases of pertussis before that. And we see that only 20 years onwards, we start experiencing a resurgence of pertussis. And after that, we have this steady increase of cases where there was actually a massive outbreak in 2012. We also see that the vaccine was introduced in 1953, and the uptake increased fairly quickly after that, with some minor oscillations in vaccine uptake in the early 70s and then in the 90s because of vaccine scares. And the other thing that is to note is that this wholesale vaccine, a vaccine composed by about 3,000 antigens, was in play nationally until the end of 2004. And in 2005, there was a complete change to an acellular vaccine, a much simpler vaccine composed by 3 antigens only. So accompanying this increase in reported cases, we actually also see changes in the allelotype frequency. So here allelotypes are a combination of alleles that give rise to different ways of entering and subsisting of the pathogen within the host. So basically they code for cell surface antigens. So what we see here is that in the early part, in the pre-vaccine era, we have a few allelotypes registered in the population, but with the introduction of vaccination, we actually see this shift in allelotype frequency over time, where some of the ones that were more prevalent in the pre-vaccine era either go eradicated or are so low in prevalence that they're very seldomly recorded. And we see that by the end of this time series in this plot, by 2018, we actually have very different population of bacteria in the Netherlands at that time. So this shift in strain frequency provides perhaps the most compelling evidence of selective forces at play. And some colleagues have, in the public health in the Netherlands, have started referring to this as vaccine-driven evolution in action, although I think that not until we started truly quantifying this has been formally tested. So what did I want to know from this Netherlands data? Is the observed resurgence a consequence of more virulent alleles? Do we see effects of vaccine changes? Do we actually see a true increase in transmission, or is it a case, as we've heard so many times now, of the more you look, the more you find? We want to understand also whether there is vaccine immune evasion. And through that, I wanted to assess the pathogen's ecological history that perhaps we could see imprinted in the underlying phylogeny. So for that, we set out to identify drivers of selections in the Netherlands, and those included vaccine changes, demographic changes, and of course, changes in transmission. So the question here is, can we spot evolution that is meaningful in the epidemiological timescale? So for that, we tend to use, in this approach, integrative parallel data stream, where we use equally surveillance data, genomic surveillance data, mathematical models within a demography heterogeneous context. So for this study, we used whole genome sequences that were isolated in the Netherlands between 1949 and 2018. And because bacteria genome are very large, we extracted single nucleotide polymorphisms using TOHAMA1 as the reference genome, simply to make the analysis a little bit more tractable, knowing all along that perhaps some of the diversity that is not expressed in single nucleotide polymorphisms could perhaps be missed. So in this analysis, we use a coalescent model. And because I'm not sure that everybody knows what the coalescent model is, I'm going to give you a very brief, less than a minute crash course on it. So the coalescent estimates population genetic parameters that are associated with coalescent events. So what I mean by this is that we start in the present and we work our way backwards in time in a stochastic process, in a random process that generates genealogies. So if we start here at time 0, A, B, C, D, and it generates a genealogy working itself backwards. So this coalescent model allows us to estimate effective population size. Basically, it's a measure of genetic diversity, and it can be seen as the number of genomes that give rise to the next generation. So the process, as I mentioned, starts at time 0 and it works itself backwards. And in the end, we are able to calculate a probability of a genealogy given the genomic data that we have. So I want to apologize that I have a small, well, not so small, but very young puppy, Great Dane, and she might be barking every once in a while because she is not fully educated just yet. So I apologize if you hear any like barking in the background. So back to the coalescent. So this coalescent, as I mentioned, is a stochastic process. And we use this continuous time Markov chain model to use molecular data to estimate these hidden genealogies. So here we see the phylogenetic reconstruction of Bordetella pertussis in the Netherlands. So the colors of the tips are for reference, where we see that yellow is pre-vaccine, whole cell vaccine is in red, so that more complex vaccine, and the acellular vaccine is in green. So for the most part, we see that there's kind of a very nice evidence of a kind of diversity decreasing over time along the trunk of our tree. And we see that for the most part, we can start understanding that there might be a decrease in genetic diversity over time. So if we see that up until the 90s, which is where the division is on the right, we see that it's mostly dominated by a few virulence factors, which I'll explain in the next slide. And then after that 1990s break, we actually see a decrease in diversity that is very, very marked in the population. So at the end of the talk, if you guys have any specific questions regarding how I do the reconstruction of the phylogeny, which I'll get into a few details a few slides ahead, but if you want to understand more specific details on the priors that I use and so on, we can discuss that, of course. So as you saw in the tree, there was this marked decrease in diversity over time. So there seems to be a clear footprint of evolution that is also reflected phenotypically when we look at the proteins expressed. And these virulence factors are responsible for invasion and persistence of the bacteria in the human host. So as you can see, through time, we start seeing this change in allele composition in the population, where some of these alleles become dominant, and they reflect this idea of a selective sweep and suddenly a fixation of these alleles in the population that seem to be a lot more fit in a highly vaccinated population. What we don't understand just yet are the immunological and epidemiological consequences of these selective sweeps. So we wanted to understand that by looking at the kinetics of genetic diversity through time using the effective population size, as I mentioned before, that number of genomes that give rise to the next generation, by looking at it as a trajectory in time, where we could then take those time series and look for association with other time sampled covariates that we were interested in. So we are using a Bayesian framework that has that coalescent-based model in the background to infer past population dynamics from molecular data. So specifically, I'm using non-parametric SkyGrid models that use a Gaussian Markov random field. And all of this is just to tell you that I smooth my trajectory so it's not too noisy. And we're truly able to compare an association with external time series. So with this, we estimate effective population size as a piecewise constant function. What I mean by this is that we take our trajectory and we define breakpoints that actually match with the sampling times of our covariates of interest. So imagine if I have vaccine data, vaccine coverage data annually, I break down my effective population size trajectory in these fixed points annually, and I observe whether there were changes in those breakpoints. So we do this by incorporating time series in a generalized linear model framework where the effective population size is a log linear function of our covariates, and we can actually estimate a posterior mean covariate effect size, so we can actually evaluate whether these associations are significant or not with a 95 Bayesian credibility interval. So I also wanted to kind of tell you within that story, of course, because we're interested in trying to understand the effects of long-term vaccination, it would actually make no point of looking at the whole genome as a whole piece. So we wanted to zoom in and see whether there were parts of the genome that the vaccine might be targeting specifically and perhaps exerting stronger selection pressures. So we took our SNPs and we mapped those SNPs to functions ranging from virulence, cell surface, transportation, regulation, biosynthesis. And we did this using two genomes of reference, the Tohama that I mentioned briefly, but also a newer one to see how that actually could change if we were to use a different reference. So here we have the calculations of effective population size. So I'm going to walk you through this because it's a large figure. So in the x-axis, we have time, and in the y-axis, we have effective population size in the log form. And the axes are the same for all of these tiny figures. And then we see that we have the genome divided into these putative functions ranging from biosynthesis to cell RNA, cell surface, metabolic, and so on, and virulence. And they're all divided. And the little tiny pie charts here refer to the amount of SNPs that are allocated per putative function. So you see that not many SNPs are observed in cell RNA. A lot are in cell surface, which is expected. Metabolic some in virulence, and some in biosynthesis, and so on. So we see that different parts of the genome actually have different numbers of SNPs that contribute to the overall diversity. The other thing that we see here is that overall, we see a decrease in effective population size, which means that we actually are seeing a decrease in genetic diversity over time. So if we look at biosynthesis, cell surface, metabolic, and virulence are parts of the genome. So the blue, the yellow, the light salmon, and the brown, these are actually located in the same area of the genome in which was experimentally observed that actually changes very quickly with environmental pressures. And the cell surface genes here are targeted by vaccines because they allow adhesion of the bacteria to the host. So we see for the most part that we do see this decrease in genetic diversity, but all along we still see some diversity maintained in the population. So I wanted to kind of show you one that was very nice and intuitive, was the metabolic decrease in diversity. And we've seen this with E. coli as well, that as a pathogen has a certain type of environment that allows it to evolve and become more reliant on their host, they actually tend to streamline their metabolic machinery such that they have to invest less energy in this part of their production as such, and invest in other parts of their genome. So we start seeing that very clearly in the metabolism. So what we wanted to do here was to try and understand whether we could group and cluster these parts of the genome in similar types of evolution. So then we can actually test, come up with hypotheses and test them, how they might be affected by these different types of covariance. So for this, we used multidimensional scaling, where it does a 3-to-3 pairwise distance and visualizes relationships. And we see three very clear clusters. One cluster that looks at biosynthesis, regulation, and virulence, and another cluster that looks at cell surface and metabolism, and then the rest of those. So that kind of allows us to think maybe these parts of the genome are affected by similar covariance. But what is very, very clear is that there seems to be some alleles that are more selectively advantageous in highly vaccinated populations. So we also, kind of a model-free evaluation, we looked at the Shannon entropy of these putative function regions to see if we also saw a decreased signal in genetic diversity. So here the light is pre-2005, and why 2005 is when we actually implement the new vaccine, and dark is post-2005. And like our estimations of the coalescence, we actually see this marked decrease for the most part in genetic diversity over time. As we evaluate the changes in entropy, they're all negative, and of course some more negative than others. But we also see that there are some regions of the genome that seem to be more varied. So on to the testing of the potential covariance. So there were three that we were very interested, that we were very interested in understanding. So how does this relate with epidemiological measures such as case reports, so annual cases of pertussis? Can we understand whether the unvaccinated births has anything to do with increased or decreased diversity? And unvaccinated births has two different types of measures here. So we know that individuals are born susceptible, and only when they are vaccinated, they become protected for pertussis. So by looking at unvaccinated births, we're not only looking at the number of individuals that are protected, but also in the context of changes in demography over time in terms of birth rates. And then finally, to test whether vaccine composition had any sort of influence in genetic diversity over time. So when we first analyzed the association between annual cases and genetic diversity, we saw that there was an association between the cases of pertussis and increased genetic diversity in virulence, regulation, and biosynthesis areas. So here on the x-axis, we have our effective posterior calculation. And remember, if it crosses 0, it's not significant. But if it's on either side, we have an association, either positive or negative. So we see that perhaps there might be evidence for increased pathogenicity or increased virulence because we see that as we have more cases, we are now seeing an explosion of diversity in these areas of the genome. When we look at unvaccinated births, like I mentioned, we're testing the effects of vaccination in the context of changing birth rates. We see that as we decrease the vaccination, so increase the possibility of transmission, we actually have an increase in diversity in the cell surface. So this for us was very exciting because we almost saw an immediate effect of relaxation of vaccine, allowing for transmission events and allowing for replication cycles to occur and perhaps facilitate transmission efficiency. And finally, vaccine composition was the last one we wanted to understand. And actually, we don't see any effect on the changes between one vaccine and the other in terms of changes in the genetic diversity over time. So what can we infer here? Based on the generalized linear models within the coalescent, we can perhaps say that there's some increased pathogenicity. There's a reduction in diversity, perhaps due to clonal expansion, because we see that there's this increased fitness in these environments. And we also see in that, although descending, but with some variation over time, that there are episodes of expansion, perhaps due to balancing selection or even immigration, because we're assuming in this model that the Netherlands population is closed, and of course it is not. And then finally, I think the most interesting one is, do we actually see decrease or increase in transmission that might be offset by the increase in surveillance? So that's where we are going next. We wanted to interrogate whether this genetic diversity was truly reflective of increased transmission. So early in the talk, I said that in the pre-vaccine era, mean age of infection was between 4 and 6. So that's one of the things that we are going to look at. So epidemiological theory tells us that if transmission is increasing, we should expect mean age of infection to decrease, so go down in terms of getting infected earlier, and the interepidemic period, so the time between epidemics, should also decrease. So if we're looking at the mean age of infection, we actually have to look at age structure data. So here we have a sample of the data between 1997 and 2018, so 20 years of data. Actually, yep. And then we see that from colors, we go from dark to light, so from young age classes to older age classes. And in the orange is the calculation of age of infection, which is a function of the transmission potential of the pathogen. So we see that even in the wholesale vaccine era and all through the transition to the new vaccine, we actually see an increase in mean age of infection, which gives us an idea that perhaps transmission is decreasing. But then some of you might be paying attention and perhaps have a more in-depth understanding of these kinds of models. and assume perhaps that perhaps we are calculating age of infection more towards an older age calculation because we're now have better surveillance than we did 20 years ago, and we might be catching these older individuals that could be skewing the population. So it was also important to look at the interepidemic period. So interepidemic period, I'm giving you a quick example that in a population without vaccination, where we set the interepidemic period to be roughly 3 years, we see the buildup of susceptibles in orange, and then we have the infection curve that has that interepidemic period. When we introduce vaccination, what we're doing is decreasing the availability of susceptibles in the population. So that is the susceptible curve. So it takes a lot more time for enough susceptibles to be available for an epidemic to occur. And so what happens is that the epidemic period becomes longer because it takes more time. So what do we see in our Netherlands population? So here we did a wavelet analysis of the surveillance data, and we're calculating the period, the dominant period of these epidemics. And we see that with time, we're now averaging about just a little bit less than 4 years by every peak, big peak. So again, we see that the interepidemic period is increasing. So together, these two are actually telling us that transmission is perhaps decreasing, meaning that the vaccine is actually doing a good job. So why are case reports still so high? So that is one of the questions that we're now trying to understand by doing a fully integrated mathematical model to analyze different sets of hypotheses, which I'm not presenting today, but if you guys are interested in this, hopefully at some point I can report back and tell you a little bit more on that. But the question now goes to the why shouldn't we expect changes in vaccine to actually contribute to a resurgence in this population? So for this, I thought I'd give you a kind of a mathematical description of why we shouldn't expect that. So we did a simulation study where we divided the population into 18 age classes, so 1 through 18. And we have a compartmental deterministic model here because we're only interested in the mechanism where individuals are susceptible. And if they encounter an infectious individual, they become infectious and then they recover at some rate. And then they can be vaccinated either by the whole cell vaccine or the acellular vaccine, depending where we are in time. So this model was calibrated with the demography of the Netherlands and also with surveys of how individuals at different age classes contacted each other and between age classes. So it's age structured with vaccine changes, trying to understand how risk changes with age over time. So because I am a nerd, I always like to show what's under the hood. And these are the equations governing the transmission dynamics. And of course, we can look at it during the question and answer session if you're interested in the nuts and bolts of this model. So if we were to do a serological study, so imagine if we took a stratification of the serology of our population, something that we're now hearing more and more, especially for COVID. Imagine we can take a slice in time, looking at what is the immune profile of the population at a given time, either whether they were infected or vaccinated. So in the pre-vaccine era, we see, again, of course, x-axis time, y-axis is the proportion of individuals in the population for a given age class that are immune. And then as before, dark to light, young to old. So in the pre-vaccine era, of course, all of the individuals that are immune would have had to have pertussis as a disease. And we see that most of the population that belong to this profile are, of course, older individuals that had disease early on in their lives. If we fast forward in time, after we introduce the whole cell vaccine, we now have two subpopulations in terms of immune profile. We now have still a lot of the older age categories which would have had the actual disease, and then the younger age categories, and some old, of course, that would have been vaccinated. And then finally, if we fast forward a little bit more, we now have three subpopulations where very few have now the acellular vaccine, mostly the young ones, and then we have the three types of vaccine profile and disease profile. So the question is here, as pathogens evolve, we should expect the population's susceptibility to change, right? So if we have this previous circulating vaccine and the pathogen, we have a population immunity to that strain, either through disease or through vaccine. But if these variants are starting to pop up, we now have individuals that are now susceptible to the new variants. So what happens in these through these different vaccine eras, meaning these different age classes? So if we were to do an experiment where we had this variant in the population, this mutant, I like to go Hollywood on this, that is 50% cross-immune to the vaccine or to people who had the disease, we can have a different type of outcome. So here, the R effective is a measure of fitness. It estimates the average number of secondary cases per infectious case in a population that is both immune and susceptible to the pathogen. So in this case, if R effective is less than 1, there is less opportunity for one infection to cause a large epidemic. In fact, the epidemic will eventually decline and die out. And of course, that is behind all of the strategies that we've been hearing throughout the pandemic for COVID, that to decrease, to squash that curve is to basically decrease R effective. So going back, if we are in 1950s, before the vaccine was introduced, we see that if we have a mutant that is only 50% cross-immune, because most of the population has this specific immune profile, it's very easy to break the R effective 1 curve. So there will be an expectation that there will be a big epidemic curve depending on how large this R effective is. As we fast forward in time, we now have two subpopulations, and we now have that alone, their contribution for the epidemic does not break R effective of 1, but of course, together, there will still be an outbreak. And finally, fast forward into the present, we have a smaller contribution of the acellular vaccine. So it will actually induce a very small selection pressure on its own, also because these younger individuals have lower contact rates. So the opportunity to transmit and to give rise to new variants is a lot lower. So if we go completely insane and have a mutant that has no cross-immunity, which for pertussis is less likely, but we know that COVID might actually generate such variants in the near future, we actually see that it's very easy to break the R effective threshold. But even in this very, very specific scenario, the whole cell vaccine could still contribute to a large outbreak, but the acellular individuals alone could not do that. So what have we learned so far? We've seen that there are signatures of Bordetella pertussis evolution. We can see how there is a mapping between epidemiology and genetic diversity. And while we can see that there's an overall effect of vaccines over time, this new vaccine, even though is a lot less rich in terms of antigens, is not responsible solely for the resurgence of pertussis in the Netherlands. And with that, I thank you for listening and I welcome questions.
All All right, wow. Okay, you've got, people are clapping or signaling clapping visually. Wow, so much to think about there. So I'm gonna open the floor to any questions from, Sylvie has her hand up and Sylvie would like to ask a question. So Sylvie, take it away.
Oh, sorry, do you hear me?
Yes.
Yes. Hi, Sylvie.
Okay. Sorry, I was trying, for some reason my webcam doesn't work. Anyway, thanks for that talk. That was fascinating. I loved it from start to finish, very clear. I have a few questions, but one, I was interested in the booster vaccine that sometimes happens, particularly during pregnancy. And I remember, I think here in the US, it's normal that you get a booster vaccine. And even, at least for my pregnancy, I remember my partner would get vaccinated too. So that means that there is an increase in booster vaccines. And from what I'm aware of, in the Netherlands, you don't have that booster vaccine. So do you think that makes a difference?
Yes, I do. So you are absolutely right. In the Netherlands, the booster schedule is different, and maternal vaccination hasn't really been taken up just yet. So in terms of that, it's too soon to actually know the effect. So I'm actually involved in a study in Brazil, who are one of the first, including the UK, to implement the maternal vaccine into the schedule, to try to understand two things. One of them is whether it interferes with the regular schedule of the infant, because of that kind of waning time that is necessary for antibodies not to interfere with each other, but also to try and understand that because not many pregnant women and families are getting boosters early on in an infant's life, whether that might in fact contribute to this increase in variance and genetic diversity in very localized areas of whatever country we are looking at, for sure.
Awesome, thanks so much. Can I ask one more question?
Yes, please.
Which is, I guess, more speculative, but I'm just thinking of, now with physical isolation, everybody wears masks and stuff. There's a lot less transmission of diseases in general, and I presume that's the same for pertussis.
Yes.
So I'm just curious what you think. Are you expecting to see a change in like the genetic profile of pertussis in the coming years?
Yes, yes. So I am perversely very excited with that situation. And I think I can say that here because you guys are equally excited about these things. So two things are happening there. Kids are not going to school every day. There's a lot of homeschooling at the moment. And like you said very well, there's a decrease in opportunities for transmission because social mixing is severely decreased, not just in school ages, but in the general population at large, which will contribute to a decrease in number of pertussis cases, which we will see not just this year, like we actually seen with influenza, right? It's the same kind of pattern. But we will see two very cool things. A change in the immune profile of the population, because now these kids have fewer opportunities to get infected earlier in age. And if they do not get the vaccine, which is one thing that we're seeing now, that a lot of kids, because they're not going to school, they're postponing their vaccination and some are not getting vaccinated, we now see this kind of protracted change in the immune profile, which could lead to several things. And one of them would be a 5 to 10 year delay and then actually observing a large outbreak of pertussis in a few years. So definitely, so there are very interesting transmission dynamic consequences that we expect to see. In terms of genetic diversity, so that's even more interesting, right? Because we have this mismatch of vaccine uptake, and we also have a mismatch in states and counties that are either going to school or actually requesting students to stay home. So we're having these very heterogeneous selection pressures that, of course, at the moment are short term. But if this stays on for a while longer, because until all of the population is vaccinated, we can actually start seeing some variants popping up that might be very, very different than what we are registering so far.
Thank you.
So there's a question here from Madison.
Yes, please.
She says, what about vaccines which have multiple vaccination options? Specifically how COVID has the new mRNA and the recombinant one, would having two different vaccine strategies affect mathematical curves? Yes, so making it very hard to predict for sure. And we won't be able to calibrate those models probably for a few months, if not for a year or so. Why? Why that is? Because in clinical trials, we have a measure of how effective a vaccine is at the individual level. That actually does not give you a good measure how effective that vaccine is at the population level. So we won't know really how that actually affects transmission potential, right? Because it might be that these vaccines are good at preventing disease, but not so good at preventing transmission at the population level. So now throw in not one vaccine, but many with different types of effectiveness, different types of uptakes, and focusing on different subsets of the population in terms of age, in terms of other types of traits these populations may have that actually affect their contact rates. So we now have a very interesting jigsaw puzzle of effects that we won't be able to truly understand this year just yet. So I think there's another question. Katie, shall I read it? So yeah. Going off on Madison's question, what makes the two COVID vaccines different, or any vaccines that have two versions, or are they essentially the same, just produced by different companies? So I'm not the best person to answer that question because I am more on the kind of like, let's simulate, vaccinate, kill some people, let them die, and so on. I know that the different vaccines have different properties. We know, for instance, that the Pfizer vaccine is a 2-dosage vaccine, which means that you will not have the full benefits of the vaccine until you get the 2 dosages. So there's an increase in how protected you are as you take the second one. So if we know that there are delays in the second dosage, so it means that we might have this population that is not fully protected. So that's going to have an effect. And we know that there are some, the Johnson & Johnson, for instance, is just 1 dosage, which means that you'll get the biggest effect in terms of immediate protection by having 1 dose. But again, we'll have different vaccines in different subsets of the population nationwide that will have that issue.